Well, good afternoon, Bruce. It's great to see you. How are you?
Doing great, Richard. Doing great.
I am very excited to chat with you ever since we had our pre-call a couple weeks ago. And I got to know you and we got to kind of talk a little bit about the many things you're involved in. We were just talking for a second about your very crazy travel schedule. Go, go, go all fall. And not to be outdone, you're heading to Europe this weekend, right?
Yep. I guess I'm like a Rolling Stone, just can't stop, right?
Well, this is great. So, for folks, we're speaking with Bruce Morimoto. This is part of our ongoing series related to v-ATPase, related encephalopathy, disorders, everything kind of that you guys are focused on. Bruce, I always kind of like to start these with a little bit of a self introduction, kind of help the audience understand a little bit about you and your background, and the work that you've been doing most recently.
Absolutely.
Yeah.
So, Richard, Bruce Morimoto, I've
Mhm.
been doing CNS drug development for coming up on 30 years now,
Wow.
working mostly with small biotech companies, but I've also been on the CRO side
Mhm.
for a bit of my career as well.
Mm.
Um, Most of what I've done is sort of what I call translational medicine.
Mhm.
So bridging the basic research into clinical development. So that's sort of where my sweet spot. And I've worked mostly in neurodegenerative diseases. So Parkinson's, Alzheimer's, frontal temporal dementia, ALS.
Mm.
Those are sort of the areas that, or the therapeutic indications that I'm most familiar with.
Mhm. And from a background training perspective, Bruce, where did you receive your training?
Yeah, so I'm a true blue Bruin. Got both my bachelor's and my PhD from UCLA. Go
All right.
Bruins.
Go
Uh,
Bruins, yeah.
did a little bit of a postdoctoral fellowship. So after I got my PhD, went up to Cal, to UC Berkeley for a while before starting my career actually in academia. So I was on the faculty at Purdue University in the chemistry department.
Sure.
And then from there, moved to the dark side, the industry part, in order to really kind of fulfill what I really enjoyed, which was much more the applied research, Richard. That's
Mm.
what really kind of got me going.
I like that. Yeah, I like that. Having spent a lot of time in industry myself, I can understand how you're taking some of those theoretical or scientific methods and trying to figure out the right applications for them. So it's exciting. It's exciting for in the space. So you kind of mentioned you've got this training in neurodegenerative diseases, research, all the work that you've done. Talk me through a little bit around people maybe for the first time don't know what v-ATPase is. How can we kind of describe this protein complex, why it mutates, and sort of why it leads to so many different severe neurological disorders?
Yeah, so ATPases are really one of the basic machineries in the body. So
Mhm.
there's a set of ATPases that are used to actually make energy in the cell. The specific v-ATPase is one that's used to actually pump out protons, so an ion, out of little compartments within the cell in
Mm.
order to make them more acid. And that is really fundamentally important for the function of those organelles, those little machines within inside the cell. And so with the v-ATPase, it's interesting because they are a genetic mutation. So there's a mutation in the DNA sequence that gives rise to this protein complex.
Mm.
But unlike what we typically think about as sort of the inherited genetic mutations, this one actually happens in utero. So
Oh,
as the baby's developing, you're bombarded. We're constant. There's mistakes
sure.
when we replicate or make copies of our DNA. Mistakes happen.
Mhm.
Most of the time, like 99.9% of the time, those mistakes are corrected. For some reason, these kids with these v-ATPase mutations, they don't get corrected. And so they make defective proteins that don't function.
Mm.
And then that causes all the sort of the cascade of events that happens after that,
Mm.
which is like slowing of the development process, the seizures that end up happening in some of the kids. All of that is a consequence of a genetic mutation that happens in utero inside the pregnant mother that
Mm.
gives rise to these defective proteins.
Mm. And we know thank you. I think that's very helpful, and I think I learned something there because I even know the in utero aspect of it, so thank you for that. And then in terms of prevalence, Bruce, we know, right, I think a couple of 100 patients worldwide, tops, right? So talk to us a little bit about, how you guys are working to kind of improve on that estimation, understand the prevalence more.
Yeah, it's an ultra, ultra rare
Mm,
disease. Now, it might be more prevalent than we think
mhm,
because the only way right now that you can know for sure that you have one of these mutations is to have your DNA sequence. And DNA sequencing has come such a long way since I was in graduate school. You can now have your own genome sequence,
mm,
for a couple of $1,000. And
mm,
so I think as we sort of screen more people that have, seizures, we might actually find that this is actually more prevalent than not.
mm.
But that said, trying to find something to fix that,
Yeah,
that becomes the real challenge, right?
yeah.
Because something that's ultra, ultra rare, something at least we know what the genetic causes of this, but trying to fix that becomes very challenging.
Mm. And I know that some of the treatment development that has sort of been exercised, I guess, right? So everything from gene therapy to drug repurposing. There's other modalities too. Maybe you could talk to us a little bit about some of those or any promising lenses that I think you see within the research community's current exploration, Bruce.
Yeah, so from my experience living and breathing drug development over
Mm.
my whole
30
career,
years, right? Yeah. Yeah.
yeah, it's interesting because drug developers think of a therapeutic indication, and then we try to find treatments for that.
Mm.
I would say in the last 5 to 10 years, there's been much more of a focus on what I would call personalized medicine,
Mm.
where now we're trying to identify what is wrong with the patient in
Mhm.
order to design the treatment that will fit them. So,
Mm.
I do a lot of work in Parkinson's disease. I do a lot of work with the Michael J. Fox Foundation,
Right.
and we're really focusing on specific targets and then trying to see whether we can identify the defect in those targets in the general Parkinson's population.
Mm.
So, what is really exciting and what I think is really cool at this point in time is that we're taking some of those learnings of personalized medicine in applying them to the treatment of these ultra rare diseases. And to
Mm.
me, that interface is really where we're going to see some tremendous, breakthroughs, is what I'm hoping. Fingers crossed, Richard.
Yeah, absolutely. And is there anyone that's and you don't need to name names but you maybe talk about in general terms, is there anyone kind of really kind of vaulting that forward or taking more significant leaps right now that are exciting you?
I think that, you had mentioned gene therapy.
Right.
And I think that that is really one of the hopes. And, a type of gene therapy is something that we call, oligonucleotide based
Mm,
therapeutics. So these are making little strands of DNA that are supposed to go into the cell and then correct the defect. So there are a number of drugs that have been approved, even for CNS indications,
mm,
um, where we've gone in and taken short little segments of DNA with the proper sequence, and actually, use that to correct the defect that's in a particular person.
mhm.
So, there's ALS is a good example. There's also something called spinal muscular dystrophy
Yep.
that we now have therapies that are based on these types of genetic mutations.
Mm,
So the hope here is that we can apply that same principle, that same idea, that same thinking, and that same mechanism for drug approval
right.
for the ultra rare indications, right? Because
Mm,
we need to get these drugs approved. We
right.
need to get buy-in from FDA, from the Europeans, from the Japanese regulator. We need to get the regulators all
All
on
on board.
board and in
Yeah.
sync, right?
Yeah. Shifting a little bit to the alliance and the v-ATPase alliance and your work there. Can you talk to us a little bit about the role you're playing for the organization, Bruce?
Yeah, so I've known Luis for a number of years. So he's
Mm,
one of the co-founders of the foundation. And we actually work together at the Michael J. Fox Foundation. I'm a volunteer there. He's
yep.
actually a staff member. And we just got to talking one time. It was actually at a conference here in San Francisco.
Mm,
And, he kinda mentioned the situation with his son, and then I also do a lot of work in the oligonucleotide and the peptide area. That's sort of
mm,
the other hat I wear in terms of therapeutics. And I remember being at a conference, it was the TIDES Oligonucleotide Peptide Conference, and I remember hearing a talk by some folks that, have been working on this, antisense oligonucleotide therapeutic, where they were actually able to treat what they call an N of 1. So
mm.
here's a patient that they had a single point mutation. They were able to use what they had learned from spinal muscular dystrophy from the approval of that drug in order to tailor a particular therapeutic for that individual.
Interesting.
And since then, they've been able to replicate sort of this model of
Mm,
being able to do that. And so, you know, Luis asked me to be on the scientific advisory board in order to help kind of share some of the knowledge and learnings that I have had over my career,
sure.
some of the workings that I've been doing in the oligonucleotide space, in order to see whether we could find some solutions
Mm.
for the problem that they're trying to tackle.
Mm. And are there moments of sort of inspiration that you guys are seeing right now that you're kind of leaning into? I mean, I know we already talked a little bit about the drug repurposing, the gene therapy, but anything else that from your vantage point that's I would say just exciting you or inspiring you?
One of the things that I've learned over my career is that in order to have the best therapeutics, we need to understand basic science. I mean,
Mm,
I'm a firm believer that basic science is where we have to start with the foundation.
mhm.
And for the v-ATPase, this basic foundation is we now have the the structure. So we know the three-dimensional structure of the v-ATPase complex. And so this is really exciting because now we can then go back to this structure, pinpoint where the defects are in these individual kids,
Yeah.
and then ask the question, those point mutations, those, defects, what is the consequence of them from a functional point of view?
Okay.
Because then that will start giving us some insight in terms of what it is that we need to do to kind of fix the problem.
Mm, mm. When I first started my career, I was working in heart rhythm disorders, electrophysiology, and started to your point, really start to have a real appreciation for basic scientists and what was being done in that general science or basic science research. And not full and being young and inexperienced. Now I look back and really realize that so much of the fundamental advancements that we were making as it pertained to cardiac pacing, guide wires, all of that stuff all came back
Yeah.
to strong basic science research, right?
Yeah, and,
Yeah.
ion channels
Mhm.
and all of those types of things, I mean, those all feed right into sort of that basic research that you are doing that has led to therapeutics for arrhythmias and things like that, right? I mean,
Right.
so we have to start from someplace, and the basic foundation is so key.
Yeah, absolutely. So maybe to that point, so the basic foundation, talk to me a little bit about some of the roadblocks, I guess, that you're I mean, funding I know is always gonna be a roadblock, right? Resources are always gonna be a roadblock. But I guess ultimately maybe the unexpected hurdles, maybe better term to use, Bruce, than roadblock, that you've kind of found, that you've come up against. And maybe talk a little bit about some of the ways you got over them.
Yeah. No, and I think that, so as a basic scientist, we're always told that you try to simplify the system as best as you can.
Mhm.
And the perfect experiment is one which you have everything controlled and you change one thing.
Yeah.
Well, biology and Mother Nature are not so kind to us.
Yeah.
Trust me,
They
Richard. I
don't
mean,
wanna help you with that. Yeah.
things are much more complex. There are many more interactions that we're learning every day.
Right.
And that's the whole reason why basic research is so exciting because you might have a hypothesis or an idea how something works. You design an experiment to test that. And
Mm.
then you let the results of that experiment experiment tell you, "Oh, yeah, you were right," or "You were sort of right, but there's something that we can't explain, and so then we need to go and design another experiment to try to figure out this unexplained part." And I think that
Mm,
with the v-ATPase, we know essentially what it does, but we don't
yeah,
know what the consequences of it. We don't know what's downstream. That's where some of the
right.
basic biology needs to
Mm,
evolve. We need our the evolution of our thinking of how these pathways work,
mhm,
and how they're regulated. I think that is sort of really the area that we need to push into in
yeah.
order to start designing better therapeutics, right?
Yeah. And again, in terms of those, I guess, unexpected hurdles or underestimations, does it kind of fundamentally go back to the basic science, Bruce, of like, look, we are underestimating this when it comes to these ultra rare neurological disorders, and we it is the basic science that we need to lean into. Was that a fair statement or
Yeah, I think that rather than hurdles, I think scientists love challenges, right? I
right?
mean,
Yeah,
if it
that's
was
why they're
simple
scientists.
and
Yeah.
if it was simple and easy, it wouldn't be fun, right? I mean,
Yeah.
it's the unexpected result that you get.
Yeah.
And, having a trained, open mind to go, okay, scratching your head, thinking about it, putting your head together, collaborating with other individuals who
Yeah.
have a different experience. That's where the real fun and power of science comes in.
Mm,
So I can see that the complexity is really a challenge for us, but this is where the ultra rares can learn from the bigger indications and
right.
vice versa. It's a
Yeah.
two-way street. We
Mm,
are learning from each other completely. A lot of what we know in terms of mechanisms for Parkinson's disease comes from rare mutations in certain
mm,
proteins of
mm.
people that display Parkinson's-like symptoms.
Mhm.
And we go, "Oh, they
Oh,
have a mutation in this particular
yeah,
protein, and that's involved in endolysosomal function or
sure.
mitochondria." And so that kind of points us in the direction of where we should be looking for the bigger Parkinson's disease by learning from sort of the more rare mutations.
Fair statement, yeah. In terms of you mentioned the collaboration, and we know how important collaboration is, right? But we also know that sometimes the collaboration isn't easy, right? It's complicated. There's sort of this connection that needs to happen, and someone needs to kind of drive that connection forward. And look, sometimes people aren't willing to collaborate, but I have seen in the rare disease community, there is more intention to collaborate, right? So, what have kind of been your experiences there, Bruce?
I think that more often than not, as long as you can get alignment of people in terms of what my objectives are,
Hmm.
what my expectations are,
Hmm.
I'm a firm believer that humans are we are all looking to do the right thing,
Right.
and that you point us in the right direction, you bring the right group together, and we put our heads together. If we're like-minded, we're gonna move forward in order to find a solution to that, and I think it requires also good leadership, right? I mean,
Mm.
you need a good captain that can bring the crew together in order to work together, right? Um, and that is a skill set that, is very important. And if
Yeah.
you could have that right person who's a leader, a cheerleader, a taskmaster sometimes, having that in an individual or maybe a few individuals that can bring a group together to work together toward the
Mm.
same goal, and that we know that we need to put our egos aside every now
Yeah.
and then, myself
Yeah.
included. It's just like, okay,
Very important.
if
Yep.
I am wrong, let's kind of like then figure out, how to make this right, right? I
Yeah.
mean, I think that that is key too.
Yeah. And from a clinical perspective, Bruce, right? Like how much do you personally get to interact with clinicians? I mean, we're talking about the researchers here, the scientists. Where's your sort of interaction point with clinicians providing care?
Another thing that I've learned through my career in doing drug development is we have to not lose sight of what our goal is. It's going
Mm.
back to having that common mission.
Mm.
And in addition to the clinician, I think I always try to remind myself that I'm not treating a disease necessarily, I'm
Yeah.
treating a patient,
Mhm.
and
Mhm.
keeping the patient in mind, what's important to them,
Mm.
and then that patient, the caregiver, the family, and the clinician,
Mm.
that forms a team of
Mhm.
people looking to help the patient itself, right? I mean, the
Yeah.
patient has to have this network around them,
Yeah.
and then I need to learn from them what's important and how to interact with them. And then, it's again a two-way street. As a researcher, I'm like, hey, there's this new development. And
Yeah.
then being able to explain it to them in terms of, okay, well, what does that mean for them? What impact? What
Mhm.
kind of clinical trial, what to expect if you enter into a clinical trial, right? I mean,
Yeah.
it's a commitment.
Yeah, for sure, right? For everybody involved.
Yeah.
Yeah. And you said you mentioned you've got a lot of CRO experience and stuff. If you were designing a clinical trial today for something, I mean, what would be, I guess, this is kind of an interesting question, I think. What would kind of be your approach in this particular instance of the ATPase?
I think that, one of the things that the Alliance is doing is collecting a lot of data, what we call, historical data
Mhm.
about the patient and the patient journey. Because
Mhm.
we know that every patient, even in some like Alzheimer's or Parkinson's, their personal experience is going to be very different. There's going to be some commonalities, but everybody's journey is very different in terms of
Right.
what kind of problems do they have? What's important to them on a daily basis?
Mm.
And so I think plotting out and understanding collectively in the rare disease space of what is that patient journey
Yeah.
becomes important because then when you go to design a clinical trial, we take into consideration, oh, this person has problems sleeping, or
Yeah.
walking is a problem, or speaking is a problem, or eating is a problem.
Mm.
And so understanding what the problem is in order to design a trial that's now doing an assessment
Mm.
of, is my drug improving the things that are important to the patient themselves, right?
Is it easy to forget that, Bruce? Is it easy? Yeah, because what you're describing to me, like, is this holistic sort of approach, right? And in the spirit of time, in the spirit of speed, is it easy to forget that? Do you have to? Do you find like you need to remind yourself that we have to look at this in through that sort of holistic lens?
Absolutely. I have
Yeah.
to remind myself all the time because not only do we have the patient and the caregiver and the treating doctor as a stakeholder, but we also have the regulatory authorities
Yeah.
as also another stakeholder.
Very good point. Yeah.
And and some 90% of the time they're aligned,
Mhm.
but we have to remember that the regulators are thinking of things in the context of risk benefit.
Right.
What is the safety versus the benefit to the
Hmm.
patient? Because
Hmm.
they're thinking of things much more in terms of, bigger populations.
Sure.
Um, and so that becomes try to balance that, okay, I need to be able to design a clinical trial where I can enroll enough people so that we have some confidence that when we get the results that they're real and that, it's not just a fluke.
Yeah.
We need to be able to replicate that result. And then having that at the same time as balancing that with what is the real need of the patient is, sort of the multi factors that we go into
Mm.
designing a particular clinical trial.
Mm. Wow. So many complicated in complex pieces to this, right? It's exciting, and like you say, scientists love to solve challenges or problems, and this is a big one. Um, in the spirit of sort of what you're doing today, Bruce, and what you hope to be doing tomorrow, from a visionary perspective, and maybe what's standing in your way, I would love to hear, kind of what you're doing today, what you hope to be doing tomorrow, and really right now, what is kind of standing in the way between today and tomorrow.
Yeah, so, as I mentioned on the Alliance side, we're collecting data on
Mhm.
individuals who have been diagnosed with this disorder. So having that kind of longitudinal data, sort of
Yeah.
the patient is here, how are they progressing 6 months, 9 months, 12 months, a
Mhm,
year, 2 years
mhm.
out? That really serves as a foundation.
Sure.
The other thing that the Alliance is doing is collecting biological samples. As I
Mm,
mentioned,
mhm,
basic biology is so
mhm,
important.
mhm.
So they have a strong effort of collecting blood and DNA from all these patients so that then we have a biorepository of
Right.
samples. So then we can match the biological samples with the patient characteristics to see if we can start finding some correlations.
Yeah.
Um, and then, once we have an understanding again of sort of the what the biological consequences of these mutations are, we can start thinking about repurposing drugs, think about the gene therapy, think about these oligonucleotide therapeutics. I think that that then becomes sort of the next step.
Mm.
So then your question is, okay, well, what is a barrier? I think
Yeah,
it
what
really is awareness. And that's why
yeah,
I was really excited when you reached out to me about this podcast because I think there are, and as I mentioned earlier, I think that there's a lot of individuals that have these types of mutations that they just don't know about it,
sure.
or that they don't know that there are these support groups out there.
Right.
The worst thing is being alone. I mean, alone and without hope, I think is the worst situation to be in.
Terrible combination. Yeah, absolutely.
And so if we can start to, develop sort of a community, which, this podcast kind of feeds into, and to start expanding that community to bring more individuals in, to get a bigger set of data that we can, as researchers, can start looking at and to
Mm,
picking through and to start thinking about. Then I think that we will start moving exponentially. It is really like that stone at the top of that rock at the top of the hill. It starts picking up momentum the further it goes down. The more data we have, the speed at which that rock is gonna be moving is gonna accelerate, and that's when we're gonna start seeing some breakthroughs.
yeah, just like you and your travel, right? The Rolling Stone. All comes back to that.
Oh, full
Um
circle, yep.
I find what's so interesting about folks like you, Bruce, is you just play all of these. You can't play a singular role in this, right? And maybe that's just indicative of sort of startup culture or in general. This alliance isn't very old, and then the work you're doing, you have to be a researcher, you have to be an advocate, you have to be a connector, you have to. To me, it's fascinating. Do you hope to get to a point where people are a little bit more swim laned, or do you think that it's really important for people to continue to have that sort of cross functional uh, uh, role? I guess.
Yeah, I mean, I can speak for myself. I mean, my
Sure.
personality, what makes me wake up and get out of bed in the morning is this passion to do something, right? I
Mm.
mean, it's funny, my whole family is like that. None of us I've got a brother and a sister, and even my mom, who's 93 years old.
God bless
We
her.
can we cannot sit still.
Yeah.
It must be something genetically wired in us, but all of us, I mean, we're constantly moving. We're
Yeah.
passionate about what we do,
Mm.
and it's that drive that makes us start asking these questions and actually to get involved. So, the
Right.
advocacy part, getting involved, volunteering, fundraising, doing the research, thinking about these problems, trying to find solutions to these problems. You
Sure.
know, that passion, I think, is something that we find really prevalent in these rare disease communities. It's like,
Yeah.
you know, we see the problem in front of us.
Yeah.
We see that there's the possibility of hope and of a solution.
Mm.
And so then it's just a matter of directing our energy and effort all combined together into that direction to kind of find these types of solutions. So
Yeah.
I
Yeah.
think that at some point in time, yeah, you're going to need experts and specialists in all the different areas, which I think is what you call your swim lanes.
Yeah.
But for now, it's fun to just wear these different hats
Yeah.
every single day, right?
Yeah,
Or
I
yeah,
must. I can imagine it is fun. So speaking of hope, and maybe we'll try to end here, Bruce. It's 5 years from now, right? And when you're looking about what you hope that the v-ATP ACE Alliance has achieved 5 years from now, patient care, research infrastructure, treatment availability, what are what are the hopes for you, I guess, Bruce?
I think, caregiver support, right? I
Mhm.
mean, I was a parent, and what was really helpful for me is to be able to share, "Hey, my kid's not sleeping,"
Yeah.
and having a common network or group to kinda share what you're going through so that you're not alone. I think so the sort of the caregiver part.
Mhm, mhm,
I think
mhm.
that the advocacy part is also important. So, getting the word out there, maybe making this from an ultra rare to a rare disease because now we start to build the community and we go, "Hey, we
Mhm.
now know that there's a lot more of us out there with these types of mutations so that we can then pool our resources because, you know, there's the saying strength in numbers, right? I
Right.
mean, there is that. And even if this was an ultra rare, be able to partner with other ultra rare or rare organizations to kind of pull our resources to share sort of our learnings. That is also really, really cool.
Yeah.
And then the third thing is really, the a better understanding of the research so that we can then, maybe take things that are happening in the Parkinson's or the Alzheimer's or the
Right.
ALS world and try to use some of their learnings once we understand our mechanism a lot better. So I think
Mhm.
it's all of that put together, so that we have some potential therapeutics that, are being tested. I think that to me would be, you know,
Huge.
all
Yeah.
right, we've been successful. We are successful.
Yeah, that's great. Folks, we've been speaking with Dr. Bruce Morimoto of the v-ATP ACE Alliance. And let's see, to dial that up, we need to do v-ATPASE Alliance.org. That's v-ATPASE Alliance.org. Once again, Bruce, thank you. Thank you for taking the time between your travels and everything else you have going on between your multiple day jobs and coming on and speaking to us about this really important work that you're doing and contributing to what we think is a really important series highlighting the ATP ACE Alliance and the disorder. So once again, thank you, Bruce. Really appreciate it.
Thank you, Richard. Fantastic.